So in this chapter, we will be discussing cell division, cell theory, who discovered them, different types of cells, as well as the structure of the cell. We'll be talking about this plasma membrane, the cytoplasm, the nucleus. We'll be going into detail about the different cell organelles like endoplasmic reticulum and Golgi apparatus. Just a reminder that all the information in this video is from the grade 9 NCERT textbook and has been either simplified or extended me. Now we know that Robert Hooke was the one who coined the term cell after looking at a slice of cork through a microscope. He coined this term in 1665 after looking at the little compartments of the dead cork cells. But then what happened after that? So after that, in 1674, Leeuwenhoek was the one who found the first free living cells in pond water. And then after that, in 1831, Robert Brown was the one who discovered the nucleus. Then in 1839, Purkinje coined the term protoplasm. Now this is when our cell theory was presented by two biologists, Schleiden in 1838 and Schwann in 1839. This theory states that 1. All plants and animals are made up of cells, and 2. Cells are the basic unit of life. In 1855, Virchow was the one who further added on to that theory. He said that all cells arise from pre-existing cells. So now the three main points of the cell theory that we can understand are 1. All living organisms are made up of cells or the product of the cells. 2. Cells are the fundamental building block of tissues, cells, organs, or any functioning organism. And 3. New cells are formed through division in pre-existing cells. So now there are two types of organisms in terms of number of cells. That is unicellular organisms and multicellular organisms. So unicellular organisms, you can tell by the name, organisms with only one cell. So this could be, there are so many, amoeba, chlamydomonas, paramecium, lots of bacteria, while multicellular organisms are those with multiple cells. So you can say lots of plants, animals, even we humans ourselves are multicellular organisms. So even though unicellular organisms like amoeba have only one cell, the division of labor in the organism is still present, just like in a multicellular organism. Just like how multiple types of cells in our body take up different tasks, like how the cardiac muscle cells help to pump our blood, and our neurons help to transfer impulses from our brain to our body and back. These kind of functions are still present in unicellular organisms, but I will get to that later when we talk a little bit more in detail about unicellular organisms. So another thing that you should remember is that the size of an organism has no relation to the size of its cells. This means, for example, if you take a rat and an elephant, the size of cells of the rat and the elephant have no correlation to their height. The cells of the elephant are not necessarily bigger than the ones of the rat. The size of the cells has no correlation to the size of the actual organism. So you should remember this because this is asked in MCQs a lot too. So don't become confused. So now let's move on to the structural organization of a cell. In any cell, there are always three main components: the nucleus, cytoplasm, and plasma membrane, although these may come in different forms. So let's begin with the plasma membrane. The plasma membrane is a selectively permeable membrane. What does that mean? This means that only certain substances are allowed to go in and out of the cell, and the membrane kind of decides that which foreign materials do I allow to come from outside the cell, and what do I allow things to go from inside the cell to go out. The plasma membrane is very thin and it is a phospholipid bilayer membrane. Okay, I know that sounds like a big word, so let's just try to break that down. Phospho means phosphorus. Lipid refers to fat. It's just another word for fat. And bilayer means two layers, which means that phospholipid bilayer membrane means that it is a double membrane that is made up of both phosphorus and fat. I'll put a picture so you can understand. So this takes us to the topic of diffusion. Simply put, the movement of a substance from a region of high concentration to low concentration. A similar situation occurs in the cell when a substance piles up, any substance piles up in high quantities inside the cell. So let's take for example, if carbon dioxide started accumulating inside the cell. This would mean that inside the cell there is a very high amount of carbon dioxide, and outside of the cell there is a very low amount. So what's happening is you have a region of high concentration inside the cell and a region of low concentration outside the cell. So what the cell will try to do is to try to balance that inequality and it will start to send CO2 from inside the cell to outside the cell. So it will start diffusing. This is an example of diffusion. So now this diffusion process can also be specific to water. The movement of water molecules through a selectively permeable membrane is known as osmosis. The reason that this happens is because when there is a higher solute concentration, either inside or outside the cell, water will move to balance that concentration. So this is where we come to a very important topic, and that is hypertonic, hypotonic, and isotonic solutions. So let's break down these words too. Hypo means less, hyper means more, iso means equal, and tonic is sort of related to the tension or tonicity of a solution. It's fine. So now to understand this a little bit more, let's take a case study. Let's take a scenario. Imagine that you have a plant cell and we're gonna place it in a salt solution. So in this salt solution, salt would be the solute and water would be the solvent. Now when you do this, this could result in three cases. 1. If the solution around the cell has more water around it, has more water compared to inside of the cell, this means that the solution is very diluted. The water will then try to balance it by entering inside the cell. So if you have more water outside the cell and then less water inside the cell, some of the water will try to move inside. But the thing is, when that water is forced inside the cell, this will cause the cell to swell up. This means that that medium around it, that solution that we put it in, is a hypotonic solution because there is more water in that surrounding solution compared to the inside of the cell. And if this was happening in an animal cell, there is a possibility that the cell could burst, but that wouldn't happen in a plant cell. Could you tell why? Hello? Yeah, because a plant cell has a cell wall, and we will be looking into that more later, and we'll figure out why it wouldn't burst if it had a cell wall. Now, moving on to hypertonic solution. Now, the exact opposite of what just happened will happen. When the surrounding solution that we keep it in has a lower water concentration than inside the cell, this means that there is less water outside of the cell than inside of the cell. To balance that, again, water will instead start moving outside of the cell. This means that when water moves out, that cell will start to shrink, and the protoplasm will start to shrink away from the cell wall. Now, what is protoplasm? It is basically the combination of the cell membrane, the cytoplasm, and everything inside it. It will basically start to shrink away from the cell wall. Now, this will be known as a hypertonic solution because there is less water in the surrounding medium than inside the cell. Now, the third case will happen when there is an equal amount of water inside and outside the cell, which means the ratio of salt to water is equal both in and out of the cell, which means there's no requirement for water to be moving at all. There will be no net movement of water, and that solution is therefore known as an isotonic solution because there's already an equilibrium of water inside and outside the cell. So now we can say that diffusion is a pretty important process as well as osmosis. So now we're going to go back to our plasma membrane. We know that it's flexible, it's made up of lipids and proteins and phosphorus, but we can also only see the structure using an electron microscope because it is that tiny. Now, the flexibility of the cell membrane that I just talked about is what lets the cell capture food and other things from its outside environment. This process is known as endocytosis, and our amoeba is known for acquiring its food using this way. So, I'm going to put a diagram here, but basically, an amoeba will try to wrap its feet around the food that it's trying to capture. It's called this. I've currently forgotten it, but this is how an amoeba or similar organisms like amoeba capture its food when it's a single cell organism and it's not like us, we can just go and pick up. It basically creates its own hands to pick it up. Next, let's move on to the cell wall. The cell wall is a very rigid, hard outer covering, and it is outside of the cell membrane, and it's only present in plant cells. And I'll come to that why. The cell wall is made up of cellulose. That's a very complex substance, and it's super hard and gives the cell structural support, and it also protects it from outside elements. This is why it's only present in plants, because for us humans or any animals, we can move around to get away from harsh environments, but plants can't do that, which is why their cells have adapted to have this cell wall covering them, so that even if they're staying in one place, this harsh environment will not affect them. Now, arguably one of the most important parts of the cell is the nucleus, also known as the brain or the control center of the cell. Our nucleus also has a bilayer membrane, just like the plasma membrane, and it is also selectively permeable. It has pores that allow items to go in and out from the nucleus to the cytoplasm. Now, our nucleus contains chromosomes, which are basically rod-shaped structures which contain DNA and protein. Our DNA is responsible for organizing and constructing new cells, while protein helps in the packaging and condensation of the DNA. Now, while the cell is not in the process of dividing, meaning it's basically while the cell is going around doing its work, the DNA is not actually already in the form of chromosomes. When the cell is sitting normally doing its work, the DNA is in the form of chromatin. Now, chromatin is basically if you took a bunch of thread and threw it on the floor. That's it. That's what the DNA looks like. It's just a bunch of thread-like material that just sits around. But when it is time for the cell to divide, for cell division, which is something we will be talking about later, the DNA will finally organize itself into chromosomes, only when the cell is about to divide. Now, the nucleus has many different important functions in a cell, from dividing to form new cells and what kind of cell it will be when it matures. Now, the kind of nucleus a cell has determines whether it is a prokaryote or a eukaryotic cell. Now, you've probably heard these terms before. So, if you're not familiar with them, let's try to break down the term first. Pro means old, karyote means nucleus, and eu means new. So, prokaryote means old nucleus and eukaryote means new nucleus. You can kind of break it down like that. So, in organisms like bacteria, the nuclear membrane is actually not present. It's like if you tried to fill a balloon with slime and then you popped the balloon, so now that slime is just kind of floating around everywhere. That slime is actually our nuclear matter. So now when the nuclear matter is just floating around like that without a membrane to hold it together, that is called a nucleoid. And in prokaryotic cells, all the cell organelles around it apart from the nucleus also don't have membranes. It's just a bunch of stuff floating around inside it. So now when we talk about eukaryotes, everything is membrane bound, meaning all of these cell organelles and the nucleus are all packed in their membranes. And all these organelles are nicely floating in the cytoplasm. Now, speaking of cytoplasm, what is it? Is it just water? Does it do anything else? Let's look at it. So, the cytoplasm is 80% water. It is a sort of jelly like fluid that holds all the organelles inside the cell and gives the cell some shape. It's like, imagine a pool, imagine a circular pool, and we throw a bunch of people and floaties and tubes into it. The water in that pool is our cytoplasm, and all the people and the items in it are our cell organelles. You can see how the water holds everybody inside of it. Now, you would notice that the people can swim through the water to get to different parts of the cell. This is how our cytoplasm works. It's also a means of transport for materials inside the cell. And other than that, it also aids in metabolic processes. So the cytoplasm is not just a fluid, it has many other functions. But right now, this year, we don't have to get into too much detail about it. Now let's look at what the cytoplasm holds. The cell organelles. In eukaryotic organisms with big and complex cells like humans, many processes are going on inside the cell that need different cell organisms to carry out those different functions. So, this is different from prokaryotic organisms which don't have a lot of cell organelles, and therefore, the functions that should be done by those cell organelles are done by a very poorly organized cytoplasm. Compare it to a small cottage industry and a big factory. In a small cottage industry, a person would collect straw by themselves and weave baskets by themselves. They would collect clay on their own and then make pottery on their own. But in a big factory, there are hundreds of different machines and so many people to carry out different processes and staff departments for different functions. So that is a difference, the basic difference between prokaryotic cells and eukaryotic cells. So there are basically five main cell organelles that we will be going through in this chapter. That is endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria, and plastids. So let's begin with the endoplasmic reticulum. Okay, this is gonna sound silly, but imagine the endoplasmic reticulum as a bunch of lasagna sheets and pasta. Just lasagna sheets and spaghetti with the membranes. That is basically what it is. This membrane that covers them is actually pretty similar to the plasma membrane of the cell. Now, when we look at the endoplasmic reticulum, there are two main types. That is the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So now, why are they called rough and smooth? So rough ER, when you look at it under a microscope, it has hundreds of these dot particles stuck to it, and these particles are known as ribosomes. Now under the microscope, it gives it a rough appearance. That's why it's called rough ER. Now, what are these ribosomes? Think of it like little protein factories. They're like hundreds of factories that convert amino acids to protein and distribute them to different parts of the cell with the help of the endoplasmic reticulum. Okay, so we know what rough ER is. It's for the production of protein. So now, what is smooth ER? Is it useless? Does it sit there in the corner like your math homework? Fortunately for the cell, no. The smooth ER takes up the production of lipids, or in other words, fats. Now, the smooth ER's fats and the rough ER's proteins. They have many uses. Some help in building the cell membrane. This process is known as membrane biogenesis. And others function as enzymes or hormones and uses. So the main function of the ER is to basically act as a sort of a pasta roadway to transport proteins and other materials around the cytoplasm and the nucleus. And if you look at this image of a cell, you can see how our lasagna sheets are centered around the nucleus. to facilitate transport between the cytoplasm and the nucleus. The endoplasmic reticulum also works as a sort of a work table for the biochemical activities of the cell, because imagine you can't do your work while floating around in the cytoplasm. It's easier to do it on the ER, and that's why the ER is basically a kind of a work table for these activities to take place. Another function, he's a do-it-all. The ER also helps in detoxifying poisons and drugs in the livers of vertebrates. Now, the Golgi apparatus. If the endoplasmic reticulum is a bunch of lasagna sheets, I would say the Golgi apparatus is a stack of pita bread, because it's a system of flattened stacks, and these are called cisterns. The Golgi apparatus is also membrane bound, and its membranes are often connected to the ER. Both of their membranes are connected together, and this really adds to that complex membrane system that you find inside the cell. So now remember the proteins and the lipid factories that I talked about in the ER? The Golgi apparatus is like the delivery system for those factories. It packages, modifies, and stores those products in its vesicles, in those cistern-like vesicles. And it transports them around the cell as well as outside the cell. Now, the Golgi apparatus also helps to form lysosomes. Wait, what are lysosomes? A what? Lysosomes are basically membrane-bound sacs filled with very powerful digestive enzymes, which are made by the rough ER. Seeing this, you could call lysosomes the digestive system of the cell, because they absorb any foreign material that comes from outside the cell, and they also break down dying old cell organelles inside the cell itself. It can do this because of its super powerful enzymes. Now, these enzymes are so powerful, in fact, that they can explode and break down the whole cell if the cell gets damaged. And that's why they're called the suicide bags of the cell. Now, moving on to the most famous cell organelle that you have probably heard of. We all know the mitochondria, the powerhouse of the cell. But do you actually know what it does, or do you just laugh at science memes to understand to look smart? Mitochondria have two membranes. The outer one is porous, again, allows things to come in and out. But the inner one has a bunch of folds in it to increase the surface area for ATP generating chemical reactions. Okay, another word here, ATP. ATP basically refers to adenosine triphosphate, and this is the energy that's used by the cell to do activities. This is the energy that mitochondria, our powerhouse, makes, and that is why ATP is also known as the energy currency of our cell. A unique thing about the mitochondria actually is that it has their own DNA and ribosomes. So this means that they don't have to rely on the ER to get their protein, and they can make their own. In fact, mitochondria and plastids are the only cell organelles apart from the nucleus that have their own DNA. Again, this is also a question that could come in MCQs. Speaking of plastids, you've probably heard this term too. These are cell organelles that are only present in plant cells, and there are two types of plastids, leucoplasts and chromoplasts. Chromoplasts contain a colored pigment known as carotenoid pigment, and this is found in fruits, the flower petals, basically anywhere with color. Now, when this chromoplast contains chlorophyll, it is known as chloroplast. These chloroplasts are very important for photosynthesis, obviously, because of our chlorophyll. And chloroplasts also contain a lot of membranes, known as thylakoids, embedded in the stroma, and stroma is a sort of structure which gives support to the cell. Again, that point is not too important, but it's good to go through it. Now again, I forgot leucoplasts. Leucoplasts are kind of similar to vacuoles. They store starch, oil, and protein granules. Now that I've said vacuoles, the last organelle of this chapter is going to be vacuoles, which are basically sacs or vesicles that store basically anywhere, anything from water to cell sap in plant cells. And in plant cells, they actually can take up to 90% of the cell's volume, and it also gives support to the plant cell. They can store so many things like amino acids, sugars, organic acids, and even proteins. But in animal cells, it's a different story. In animal cells, vacuoles are usually very tiny or even non-existent. It's not very important in an animal cell to store food for a long time. In single-celled organisms like amoeba, the vacuoles are known as food vacuoles. The food vacuoles contain food that the amoeba has consumed. Remember, I had told you before about the pseudopods that go around the food and capture it. It brings it to the food vacuole, which stores it. Now, hopefully, you remember the cell theory that I talked about a few topics back. Hopefully, you haven't forgotten it. If you haven't, you would remember the third point, which is that new cells are formed by the division of pre-existing cells. So let's get into that. The process by which new cells are created is known as cell division. Now, there are two types of cell division, mitosis and meiosis. It's kind of easy to get confused in it, but you just need to try to remember the difference between them. If I had to describe it in one word, it would be like mitosis, growth, meiosis, reproduction. Now let's look at mitosis. Mitosis is when the cell divides for growth. The mother cell divides into two identical daughter cells. Now these two identical daughter cells will have the same number of chromosomes. It will not change. And I guess for us it would be usually 46 chromosomes. So in mitosis, remember that the mother cell splits into two identical cells and the number of chromosomes stays the same. This process is mainly for growth or tissue repair. Like I just said, mitosis is for growth, meiosis is for reproduction. During meiosis, the mother cell will now split into 4 different new cells. Now, this time, these new cells have half the number of chromosomes. Can you think of why? So this is because later on these cells are going to become gametes. Now gametes are the cells that will further combine to become zygotes. So there are two types of gametes, our female gamete and our male gamete. Male gamete would be sperm and female gamete would be our ovum. When these two combine, they form a zygote which later becomes a baby. Now, for that baby to be born healthy, it needs to have the same 46 number of chromosomes. So that's why male gamete 23 plus female gamete 23, 46 chromosomes. This results in proper reproduction. Okay, so that's it for this chapter. This is actually my most favorite chapter in 9th grade. I've moved on to 10th now, but I just love biology, which is why I decided to start with this chapter first. Now for biology, I'm giving you tips. You need to break down the concepts when you learn them, because this chapter and basically everything else that you learn in 9th grade, not even just science, math, social studies, everything, it's basically a foundation for whatever you're going to do in 10th. So if you want to do well in 10th grade, you need to be strong with your concepts in 9th grade. So you need to know everything like the back of your hand. Now for subjects like biology, I like to pretend that I'm teaching somebody, which is basically what I'm doing in this video. So I hope you were able to understand something from this video, and if you have any doubts or other chapters that you're interested in seeing me cover, please comment and subscribe for more class 9 and 10 chapter explanations.